Hi everyone. In this video, we are going to discuss about guidelines of Cpcsea for conducting the experiments on animal. This is Dr. K. Prabhu. In India, a huge number of animals are used for conducting various experiments and researches. Good laboratory practices for animal facilities assure quality maintenance and safety of animals while conducting biomedical and behavioral research and testing of products. Hence, the proper use of animals in various experiments and researches, the Government of India on 15th December 1998 has constituted the Committee for the Purpose of Control and Supervision of Experiment Animals, shortly called as CPCSEA. The CPCSEA has guidelines for the laboratory animal facility. It is expected that scientists use this document as the baseline information to follow procedure in their experiments involving various species. Use of animals in experiment or research. Guidelines for experiments on animals. There are four guidelines are there. CPC guidelines on the norms and practices for regulation of animals experimentation. CPC guidelines for laboratory animal facility. Third one is Indian National Academy Science Guidelines for Care and Use of Animals in Scientific Research. The fourth one is ICMR Guidelines for Use of Laboratory Animals in Medical Colleges. And there are three acts and rules are there for the experimentation on animals. The first one is the Prevention of Cruelty to Animals Act 1960. The second one is the Breeding of and Experiments on Animals Rules 1998. The Breeding of and Experiments on Animals Amendment Rules 2001. The purpose of the CPCSEA guidelines for the laboratory animal facility is to promote the human care of animals used in biomedical and behavioral research and testing with the basic objective of providing specification that will enhance animal well-being, quality in the pursuit of advancement of biological knowledge that is relevant to human and animal care and technical personnel. And adequate animal care is must we should provided by a veterinarian or a trained experienced person. Daily observation of animals should be done by someone or other than the veterinarian. The veterinarian can also contribute for reviewing protocols and proposals, animal husbandry and animal welfare, monitoring occupational health hazards, containment and zoonosis control programs, and supervising animal nutrition and sanitation. Institutions should employ people trained in laboratory animal science or provide for both formal and the on-job training to ensure the effective implementation of the program. In India, the central government offers the training courses to handle the animals. Animal procurement. All animals must be acquired as per the CPCA guidelines. Transportation method should be followed. The next part is quarantine, stabilization, and separation. Newly received animals should be quarantined from those already in the facility. A minimum duration of quarantine for small animals like a mice, rat is 1 week. For larger animals like a cat, dog, monkey is 6 weeks. Newly received animals should be given a time for physiologic, psychologic, and nutritional stabilization before we use for the research. Species separation of animals should be done to prevent disease transmission and to eliminate anxiety and possible physiological and behavioral changes. Cubicles laminar flow units, cages that have filtered air or separate ventilation, and isolators shall be suitable housings. In some cases, different species of animals can be kept in the same room. Example, if two species have similar pathogen status and are behaviorally compatible. The next topic is surveillance, diagnosis, treatment, and control of disease. When we use the animals should be observed daily for diseases. Unexpected death and a sign of illness, distress in animals should be reported promptly to ensure appropriate and timely delivery of veterinarian medical care. Animals that show signs of a contagious disease should be isolated from the healthy animals. If an entire room of animal is known or believed to be exposed to an infectious agent, the group should be kept intact and isolating during the process of diagnosis, treatment, and control. Personal hygiene. Animal care staff must maintain the personal cleanliness, such as showers, change of uniforms, footwear, etc. Clothing suitable for use should be supplied and laundered by the institution. Personnel should change clothing as often as is necessary to maintain personal hygiene. Personnel should not be permitted to eat, drink, smoke, or apply cosmetics in the animal room. Separate area or room should be used. Animal experimentation involving hazardous agents. Institution Biosafety Committee, shortly called as IBSC, look for safety issues. The Institutional Biosafety Committee shall also examine the proposal on animal experiments involving hazardous agents. The procedures and the facilities must be reviewed by both the IBSC and IAEC. Multiple surgical procedures on single animal. Multiple surgical procedures on a single animal for any experiment are not to be practiced unless specified in the protocol approved by IAEC. Duration of experiments. No animals should be used for experimentation for more than 3 years unless adequate justification is provided when the proposal is submitted into the IAEC. Physical restraint. Brief physical restraint of an animal for examination, collection of samples, and other clinical and experimental manipulation can be done manually or with the devices to minimize stress and avoid injury to animals. Prolonged restraint of any animal should be avoided unless essential to research objectives. Restraints devices cannot be used simply as a convenience. Period of restraint should be minimum. Animals to have restraint devices should be given training. Veterinary care should be provided if lesions or illness associated with restraint are observed. The physical condition and design of animal facility determined to a great extent the efficiency and the economy of their operation. The design and the size of an animal facility depend on the scope of institutional research activities, animals to be housed, relationship, to the rest of the institution and geographic location. Physical relationship of animal facility to laboratories. Good animal husbandry and human comfort and health production is required for separation of animal facility from personal areas. Example, offices, conference rooms, labs, etc. Lab animals should be housed in an isolated building located as far away from the human habitations and not exposed to dust, smoke, noise, wild rodents, insects, and birds. Animal quarters can be made in separate building, wings, floor, or room. The animal housing area should be placed adjacent to or near labs, but separated from them by barriers like entry locks, corridors, or floors. Space should be well divided for various activities. Animals' rooms should be occupy about 50 to 60% of the total constructed area in the animal house, and the remaining area should be utilized for stores, washing, office and staff, machine rooms, quarantine and corridor, etc. Since animals are very sensitive to environmental changes, sharp fluctuation in the temperature, humidity, light, sound, and ventilation should be avoided. Functional areas. Sufficient animal area is required to ensure separation of species or isolation of individual project when needed, and for providing animal housing. In small facilities, maintain few animals or maintain animals under special conditions. Some areas listed below are included in multi-purpose area. Specialized laboratories are individual areas near animal housing for surgery, intensive care, necroscopy, radiography, preparation of special diodes, experimental manipulation, treatment, and diagnostic laboratory procedures or equipment if hazardous biological, physical, or chemical agents are used. The second one is receiving and storage of areas for food, bedding, pharmaceuticals and biologics and suppliers is third one, and fourth one is space for administration, supervision, and the direction of facilities. And the fifth one is showers, sinks, lockers, and toilets for personnel. And the sixth one is an area for washing and sterilization of equipments and supplies. And seventh one is an autoclave for equipment. And eighth one is a food for bedding and separate areas for holding soiled and cleaned equipment. Then an area for repairing cages and equipment, and an area to store waste prior to incineration removal. Then the physical facilities, as we already discussed, building materials, corridors, utilities, animal room, doors, exterior windows. So these are the physical facilities should be maintained as per rules. Then the environment is also already we discussed. The proper temperature, humidity, ventilation, power and lighting and noise control. Animal husbandry. Caging or housing system in animal husbandry should be designed carefully to facilitate animal well-being, to meet research requirements and minimize experimental variables. The housing system should be following the factors: provide space that is adequate, permit freedom of movement and personal postural adjustment, and has resting place to that species, provide a comfortable environment, provide an escape-proof enclosure that confines animal safety, provide easy access to food and water, provide adequate ventilation, meet the biological needs of animals, keep the animals dry and clean, consistent with the species requirement, facilitate research while maintaining good health of animals. House should be constructed of sturdy, durable materials, polycarbonate and stainless steel cages. So, like this, these factors we should follow when we caging or housing the system in animal. Next, shelters are outdoor housing. Shelters should be accessible to all animals, have sufficient ventilation, and be designed to prevent buildup of waste materials and excessive moisture. Houses, boxes, shelves, perches, and other furnishing should be made of materials that allow cleaning or replacement. Ground level surface of outdoor housing facilities can be covered with absorbent bedding, sand, gravel, grass, or similar material. Building of animal waste and stagnant water should be avoided. Social environment. So attention should be given to whether the animals are naturally territorial or communal and whether they will be housed singly or in groups. Group housing should be considered for communal animals. Group composition should be held as stable as possible, particularly for canine, non-human primates, and other highly social mammals. Because mixing of groups or introducing the new members can alter the behavioral and physiological functions. Non-human primates should have a run for free-ranging activities. Activity cages are often used for short-term housing of dogs and may be necessary for postsurgical care, isolation of sick dogs, and metabolic studies. Pen runs or other out-of-cage space provide opportunity for exercise, and these should be used to hold dogs for long periods. Bedding. Bedding should be absorbent, free of toxic chemicals or other substances that could be injure animals or personal and of a type of not readily eaten by animals. Bedding should be removed or replaced with fresh materials to keep clean and dry. Bedding can be changed twice in a week. Food. Animals should be fed palatable, non contaminated, and nutritionally adequate food daily. The food should be available in amounts sufficient to ensure normal growth in immature animals and maintenance of normal body weight, reproduction, and lactation in adults. The food should contain adequate nutrition, including formulation and preparation, freedom from chemical and microbial contamination. Bioavailability of nutrition should be at par with the nutritional requirement of the animal. Precautions should be taken if perishable items such as meats, fruits, and vegetables are fed because these are potential source of biological and chemical contamination. Meat, fruit, vegetables, and other perishable items should be refrigerated if required to be stored. Unused open food should be stored in vermin proof condition to minimize contamination and to avoid potential spread of disease agents. Water. Animal should have continuous access of fresh, potable, uncontaminated drinking water. Periodic monitoring of microbial contamination water is necessary. Sanitation and cleanliness. Animals, rooms, corridors, storage spaces, and other areas should be cleaned with detergents or disinfectants. Cleaning utensils, example mops, pails, and brooms should not be transported between the animal rooms. Where animal waste is removed by flushing, cleaning should be done at least twice a day. Cages should be sanitized before animals are placed in. It can be done by washing solid bottom rodent cages and accessories once or twice weekly, and cages racks at least monthly. Disinfection can also be done by some chemicals. Periodic microbiologic monitoring should be done for disinfections. Rabbits, guinea pigs, and hamsters produce urine with a high concentration of protein and minerals. These compounds require the treatment of acid solution before washing. Some sterilizing equipments like autoclave gas sterilizer are essential when pathogenic organisms are present. Assessing effectiveness for sanitation. Visual inspection of materials, monitoring water temperature, or microbiologic monitoring should be done for this purpose. Waste disposal. So one of the accessing effectiveness of sanitation. All waste should be collected and disposed of in safe and sanitary manner. Pest control programs designed to prevent, control, or eliminate the presence or infestation by pests are essential. Emergency weekend or holiday care. Animals should be cared by qualified personnel every day. In emergency, institutional security personnel and fire or police officials should be able to reach people responsible for the animals. It can be enhanced by posting names or telephone number in animal facility or replacing them in a security department. Record keeping. The following should be done for recording purpose: animal house plans, animal house staff record, health record of staff and animals, standard operating procedures relevant to animals, breathing stock purchase and sales record, minutes of IAC meetings, records of experiments done on animals that should be maintained separately in Form D, death record, clinical record of sick animals, trained record of staff involved in animal activities, water analysis report. So this is the Form D format to record of animals occurred and experiments performed. It should be maintained by the investigators. So in the form, the various information like date of entry, number of animals recorded, name and address of the person authorizing the experiment, certification of the investigator authorizing the experiment. So like these details we should given. Then the personal and training. Staff must be provided with all record protection protective clothing like mask, apron, gloves, gumboots, other footwear, etc. Facilities should be provided for changeover with the lockers, wash basin, toilets, and bathrooms to maintain the personal hygiene. Transport of laboratory animals. Animals can be transported by road, rail, or air. The transport stress should be avoided, and the containers should be of an appropriate size so as to enable the animal's comfort, free movement, and protection from possible injuries. Laboratory animal ethics. All scientists working with the laboratory animals must have deep ethical consideration. The ethics are taken care at all levels, that is, at the individual, institutional, and national level. Anesthesia and Euthanasia. In painful experiments, appropriate anesthesia should be given to animals. It must also be ensured that the anesthesia is given for full duration of experiment and at no stage the animal is conscious to perceive pain during the experiment. Euthanasia is restored to events where an animal is required to be sacrificed or termination of an experiment or otherwise for ethical reasons. Investigator must ensure the animal is dead. Conditions or regulation for approval of experiments by AAC. The study must be conducted by only trained staff who is familiar with animal care, handling, and ethical issues. A register must be maintained by animal house in charge, HOD, containing information on names and biodata, and training records of scientists supporting staff who performed the set experiments with the data of study. A brief outline method procedure is adopted for the study with a source, number, sex and species, breed of animals employed for the study, and the conditions are husbandry maintained. Results of the study is collected with a copy of publishing report. Follows up the observation of record of animal after completion of the set study with the information of health status, illness, and procedure of euthanasia or rehabilitation adopted, if any. Record of reuse of animals, if any, with the details of waiting periods before new study or experiment performed. General conditions and health record of all animals that are use such studies listed above. Other provisions laid down in the CPCA guidelines. So thank you for listening.